Hey everyone, welcome back to my channel again. So I apologize for delaying to make another video. So today we're going to look at the classical pathway. I think I'll post about the alternative and lectin pathway tomorrow. So I'm going to explain the classical pathway in the best way I can explain it. So why is okay, maybe I don't know. Maybe let me just say how I understood it, why is it called the classical? Let me write this in capital letters. The classical pathway. So basically, it's the first to be discovered. It's like the oldest pathway to have been discovered, just like the way we have classic music, the classical pathway. So today we're going to look at the classical pathway, and you can be asked the first thing you need to know at the back of your mind. I hope you remember from the first video. If you've not watched it, please go and watch the introduction I gave in the first video. There are 9 complement proteins. There's C1, C2, C3, C4, C5, C6, C7, C8, and C9. So these ones don't participate in this whole cleavage process. They form what we call the MAC, which in full is the membrane attack complex. And then these ones, these other ones which I have not put in a box, participate in cleavage. Like they participate in the whole cleavage situation that um, brings about that whole cascade sort of thing. So I highlighted in the previous video that you need to take note of C1 and C2, and we're going to see why we had to take note of C1. And I said in the previous video, we shall see more of C1 in the classical pathway. So you're going to highlight this, and in the previous video, we said how C3b was an opsonin, and then we gave that the 3 main aims of complement proteins is for opsonization, inflammation, and cytolysis, or like lysis of the breakage of the membrane of this pathogen. So we're going to look at the classical pathway, and you realize that as you look at it, you're going to see how the opsonin is going to be formed, that's C3b, how the anaphylatoxins are going to be formed, which we talked about, which are respectively C3a, C, C4, I think C4a. Yeah, it's actually C4a and C5a. These are anaphylatoxins. I think if you don't know what their function is, go watch the previous video. You'll see what the importance is. So we're going to look at the classical pathway. And here I've where I'm highlighting C1 is triggered. No, you can be asked, the first question you can be asked is, what triggers this pathway? What brings about this pathway? So let me just recap a little. What triggers this pathway? Let's just look at this diagram here. Let me zoom into it. Yeah, let's just look at this diagram. Focus on this diagram here. So you see this diagram. We said, I think you can see where my cursor is moving, this area here. That's where I'm teaching from. So I think you can see that, as I told you previously, C1 is a complement protein that is made up of C1q, C1R, and C1s. And I told you in a previous video, it has 6, C1q has 6 subunits, which I think you can see here. You can count 1, 2, 3, 4, 5, 6. So this is C1q. And these are the other structures, C1q and C1s. So we said C1q is the one that's going to bind to the Fc portion. So someone may ask, what's the Fc portion? So when you look at the structure of an antibody, it's here. Now let's move to this other side. The Fc portion is this lower part here. I've drawn for you the structure of an antibody. I've not drawn it. I picked it from the internet. Sorry. This is the Fc portion. The fragment Fc in full is fragment constant. This is like a constant region. Then FAB portion is the fragment, I think antigen binding or antibody binding. antigen binding, because this is antigen binding site, yeah. So that's it. So you can be asked now, where I had to specify is that this C1q binds to the Fc portion. So you can be asked what triggers this pathway. I've highlighted it for you that C1, it is triggered when C1 binds to an Fc fragment of an antibody that is bound to an antigen. I think you can get that image in the internet. Like when you see I see one complement protein is bound to what? To an antibody that is already bound to an antigen. So remember, the function of these proteins is to complement the pathway. They enhance the pathway, they improve it. To complement someone is to make someone feel good. So it makes the thing easier. So basically, this thing is attaching to enhance this whole process. So for this cascade to happen, this C1q molecule, the one that's having the 6 subunits, remember I told you in the first video that this shows you that this complement protein is able to bind to up to 6 antibodies. But for the cascade to happen, or for like the trigger to happen, this C1q needs to be bound to at least 2 antibodies. So if it doesn't bind to at least 2 antibodies, that cascade is not going to happen. So C1 binds to the antigen. I'm now reading the next point here. C1 binds to the antigen antibody complex, which leads to C1R, which later activates C1s. So C1R activates C1s. After C1q binds to the Fc portion of the antibody, C1R is activated, which in turn activates C1s. So what happens? Let me just get another page. So we have said C1 is bound to the Fc portion or fragment of an antibody bound to an antigen. So now, that's the first point you need to note down. So C1q activates C1R, that in turn activates C1s. So C1s is going to cleave C4 and C2. These are the important first complement proteins you need to note. So the first ones we have used are C1, C4, C2. Someone may be asking, how comes we have not used C3? Because we don't have what cleaves C3 yet. So this is going to cleave C4 and cleaving means breaking down. So C4 is going to be cleaved into C4a and C4b. And we said B is the larger fragment. This is the larger fragment. And then we said C4a is an anaphylatoxin. And now I think you can see that the goals we said are being achieved. We said the goal of this complement protein is opsonization, inflammation, which is brought about by the anaphylatoxins, and we have formed SOFA1 here, and cytolysis. So you're going to see in this whole process, like in the complement pathway. So this larger fragment is going to remain. This one will be like, it will diffuse away. So you have C4b here as the larger fragment. So C2 will be also cleaved into C2a and C2b. But remember, we said C2 is a special kind of complement protein. So here, C2a is now the larger fragment. C2a is now the larger fragment. So C2a, this is not an anaphylatoxin, note that down. C2b is not an anaphylatoxin, it's not involved anywhere else in the pathway. So C4b and C2a are going to combine together to form C3 convertase. This is a very good question. They like asking in a multiple choice question, that what forms, like what makes up C3 convertase. I think you've seen it here. So now you've seen C3 convertase has been formed. That's when now the C3 is going to come in. C3 convertase is going to cleave C3 into C3A and C3B. Now here, I think you can notice that C3B has been formed. And what did it say C3B was? An opsonine. We have achieved an anaphylatoxin. We have achieved an opsonine, which is C3B. So what's going to happen next? So C3B is going to combine with these that we formed up here. Yeah? C3B is going to combine with those. This is now an anaphylatoxin, C3A. Remember we said it was one of them. So these ones are going to combine plus C3B. So it's going to have C4B, C2A, and C3B. This is what we call C5 convertase. This is what we call C5 convertase. So after we have formed C5 convertase, I think you can now know what is going to be cleaved. It's going to be C5 that is cleaved into C5A and C5B. I think you can see another anaphylatoxin has been formed. And then C5B is going to bind to those other proteins I showed you that don't participate in this whole cleavage. It's going to bind to the C6, C7, C8, C9. So C5B will bind to C6, C7, C8, C9 to form the membrane attack complex. The membrane attack complex, which will create pores, which creates pores in the membrane of the pathogen. Finally leading to lysis. So can you see that we have achieved all the goals of complement proteins? I think you can see it now. So in summary, yeah, we have C1, which cleaves. Let me just try to draw a chart diagram here. We have C1, and C1s is going to cleave C4 and C2. This will be C4A and C4B, and this will be C2A and C2B. So C4B plus C2A form C3 convertase. Yeah. Pardon my handwriting. They form C3 convertase. And then C3 convertase is going to cleave C3. This will cleave C3 into C3A and C3B. C3B is going to combine with this. So you're going to have C4B, C2A, C3B, leading to C5 convertase, which cleaves C5 into C5A and C5B. C5B binds to C6, C7, C8, C9, forming the membrane attack complex, leading to lysis. I hope this made sense for the classical pathway. I've tried to explain it the best way I could. Thank you so much for watching my video. Feel free to share, like, and subscribe.